about the bisulfite conversion, or we talk about bisulfite sequencing as well. Bisulfite sequencing is a method for the detection of DNA methylation. But before we talk about the bisulfite conversion, I would like to introduce with epigenetics. Here we have two mice, and we can already see they have a different phenotype. So the first mouse here is dark gray, and the other one is brighter in color. But what if I tell you that these mice have the same and identical DNA sequence? We know that genes are responsible for phenotypes. But how is a variation in phenotype, for example, possible if I tell you that these mice have the exact same identical DNA sequence? This is possible with epigenetics. The mice have the same DNA sequence, but different epigenetic patterns. In genetics, we talk about genes, and a mutation can alter the DNA sequence, and this can possibly lead to changes in the phenotype. However, in epigenetics, we have no alteration in the DNA sequence itself. There is not a single nucleotide changed here. However, we have a methylation of a cytosine, for example, and methylation or different other epigenetic changes, such as histone modifications, can also change the phenotype. DNA methylation is one of the most abundant phenomenons if we talk about epigenetics. In most animals, DNA methylation occurs on a cytosine, so cytosines can be methylated. Here we show a gene sequence. In green, we have the promoter region. We see that in the promoter region, we have DNA methylation, so cytosines in this region are methylated. This usually leads to the inactivation of genes. In this scenario, genes are not transcribed anymore. But how can we detect these methylation patterns? Now we speak about bisulfite conversion. So DNA methylation involves the cytosines. Some cytosines are methylated and some are not. With bisulfite conversion, the methylated cytosines will stay the same, they will stay methylated. However, the cytosines which are unmethylated will be replaced by uracil. Here again we have this cytosine and we have a methylated cytosine. With bisulfite conversion, the cytosine will change to uracil in the DNA, and the methylated cytosine is untouched by bisulfite. So after bisulfite conversion, we generate a DNA where all cytosines which are unmethylated are replaced by uracil. After certain rounds of PCR, the U will be replaced by T. So basically we can say that all unmethylated cytosines will get replaced by thymine in the end. When we compare the DNA sequence now, we can easily figure out where the methylated cytosine occurs. So all cytosines which are replaced by thymine are considered unmethylated. However, in the regions where the cytosine stays cytosine after bisulfite conversion, we know there we have DNA methylation. This knowledge can be very helpful when we analyze genomes in dis-ease. Bisulfite sequencing can help to figure out potentially silenced tumor suppressor genes, which when silenced are candidates for tumor promotion. So with bisulfite sequencing, we can exactly determine DNA methylation patterns. And with that, I thank you very much for watching.